Hello, my name is Rachel, and I'm the Research Support Coordinator at the Cure Starts Now. And today we're joined by Dr. Dasgupta from Emory University Hospital. Welcome, Dr. Dasgupta.
Hi, Rachel.
So, Dr. Dasgupta, I know the Cure Starts Now and the DIPG Collaborative have partially funded some of your work the past couple of years. And just to kick things off, can you share with us your journey from studying tumor metabolism into discovering sumoylation as a potential vulnerability in DIPG?
Yeah, hi. So my journey with Cure Starts Now, it started many years ago. Our first funding came, I don't exactly remember which year, but was probably 6, 7, 8 years ago. Uh, and we continued on that work, and as we progressed to understand the vulnerabilities of DIPG, these tumors. We um, we kind of stumbled upon this specific idea. This idea came from a different tumor, a neuroblastoma of children. And as I was reading a study, it struck me that some of the pathways that are presented in that study, I felt that some of these pathways have similarities with DIPGs based on my understanding of the disease. And the first thing we did is just, we bought these drugs that are available, and we just tested on DIPG cell lines that we obtained from our collaborators, or we generated from biopsies or autopsies. And I had never seen results like this in the lab. And during that time, 2 years ago, the project was not funded. That's how we start. Sometimes we all scientists start some experiments, our projects, when they're really not funded, and we get excited about the data, and then we apply for grants. Um, so then we repeated the experiment in multiple DIPG lines that we got from all over the world, really. And then when almost every single line responded, I thought there is something in it. And that is the time I approach Keith, I think we got something here, but this is not a funding cycle. And so, but we want to continue this research. And so what do we do? We need some funding here because I think there's something in here, and we want to explore more lines. We want to go deep to understand the mechanisms and all that, and animal studies, um, because this drug was actually in clinical trial. Is still in clinical trial in multiple cancers. This drug was a little too big to cross the blood-brain barrier, go penetrate the brain. We knew that from the beginning, but we at least wanted to test in vitro and then the cell lines. And so Keith did something remarkable, that he was able to raise that money, and we wrote a proposal, and then Keith raised that money, and that's how the work got started. I mean, trust me, I don't know, without that seed money, we would be here. Absolutely. I'm 99% sure that I will not be able to. I don't know where I will get the money from. And that little seed funding helped me fund one person, at least partially, and buy the expensive drug. The drug's really expensive. Uh, and then we moved to Emory. As you know, that I was in Cincinnati Children's, and I relocated to Emory University, and my whole lab came with me. And the person who actually worked on the project, she also came with me, Skylar. So we continued working on the project. And as we continued to work on the project, we found our collaborators here in Emory University. There were Toby McDonald, he's our head of neuro-oncology, pediatric neuro-oncology. He has expertise in medulloblastoma and DIPGs. And then I met Andy Hong, who has expertise in another lethal childhood tumor called ATRT. And so we discussed with them about this pathway, and they said that, hey, Toby and Andy, we are working on this pathway, and given the gene expression data, of medulloblastoma, especially the high-risk medulloblastomas, the group 3 and group 4 medulloblastomas, and the ATRTs, given the gene expression data, it seems like these tumors um, might also um, respond to this drug. Sumoylation probably could be important here. And so that's how we, expanded the project, and people from their lab got involved. We had joint meetings, and then Skylar actually spearheaded this collaborative project. And now we have data on over 30 cell lines between DIPG and medulloblastoma group 3 and 4, and including circulating tumor cells and ATRT cells. Every single line responded. I mean, there was not a single line that we found that did not respond. I mean, of course, there's variation, but every single line responded. And then we came across another drug that hits the same pathway, okay, and the same Sumoylation pathway. I mean, the name, I don't know what its name, the name came from, it's called Sumoylation. So the Sumoylation is a process by which small proteins in the cells, they modify other proteins, and by modifying other proteins, it mostly makes them more active, makes them the target proteins more stable. And say, for example, if some of the target proteins are oncogenes, the Sumoylation process makes those oncogenes stay active for a longer period of time. So if you block the Sumoylation process by a Sumoylation inhibitor, then those oncogenes, active oncogenes that actually cause the disease, they won't be as active for as long if they were not Sumoylated. So which means that the inhibitor is probably causes instability of this cancer-causing proteins. And so that is essentially the hypothesis. And so uh, moving here, we now, first of all, we collaborated with Toby and Andy to expand our work on beyond DIPGs. That's one. Secondly, to understand the mechanisms, how it works in these tumors, we use some of the funding to do a process called mass spectrometry proteomics. It's a technical term which essentially means to identify which genes, which proteins in the DIPGs are getting modified by the Sumoylation process. So now we have identified about 15 to 20 targets, and we now know one specific target that is very actively Sumoylated. And that is an important gene that regulates that is responsible for the genesis, we call it transcription, genesis of multiple oncogenes. And so we think that could be a target, but at the same time, I would say that the drug is potentially targeting multiple proteins and blocking their Sumoylation. And so what we have done after coming here in Emory is that we needed more funding. And therefore, we reapplied to CureStats Now, and the scientific review committee and Keith, everybody else were really kind enough to fund us again, to renew this funding. So we got $100,000 from $100K, and with this money, we actually did this proteomic study, and now we found that there is another drug that hits the same pathway, and that's a little smaller drug. Okay, the drug actually, the size is smaller, and so we thought that it would reach the brain a little better. And so we tested that drug. So that drug is not on clinical trial, but it targets the same Sumoylation pathway. And we ran 2 experiments, and it was beautiful in mouse. So it extended in the first line, the mice that were treated with DIPG, they lived for 3 times as long. In the second line we tested, it lives about 2.5 times as long. And so now we are going to test in another animal model where the animal has an intact immune system, because the patients have an immune system. So we'll test this in the mouse that has got an intact immune system. And we're going to test actually, we have another collaborator here, Tim Gershon, who's also an expert in T cell therapy, the T cell immunotherapy. So we're going to test whether this drug, this smaller drug, enhances any CAR T cell immune therapy in DIPGs. So these are all getting set up now. We are doing two other things on this project. One is how to push more drugs to the brain. So one way we are trying to push more drug to the brain is as is done in patients is injecting the drug directly into the brain, to the cerebrospinal fluid, through intracerebroventricular delivery. It's called ICV. It is done in patients. So we are doing it in mouse now. At the same time, we are using another method called ultrasound. It's called through this ultrasound method, we have optimized now how to push more drugs. So in this ultrasound method, what it does is that it creates bubbles. We actually inject bubble we inject bubble to the tail vein, and then when we apply the ultrasound in the brain area, in the brainstem area, the ultrasound makes the bubbles burst, and that temporarily breaches the blood-brain barrier, allows more drug to get in. So we have optimized this method, and we can test if this small drug called HB007, if we can push more of these drugs to this ultrasound method. The other thing that we are doing is, because none of these compounds, none of these drugs they don't belong to us, right? So we need permission from companies, and who knows, they may or may not be interested in clinical trials, right? And it's really up in the air. Sometimes, as you know, that because the patient population is really small, the target population is really small, pharma doesn't get much interest in it because their market share is really small. It's as unfortunate as it is. So, for example, if one Sumoylation drug is in lung cancer trial, they really don't want that drug to be tested in a disease like DIPG, because it's a really small population and they don't want to interfere with their current clinical trial. But for DIPG, there's urgency, so we cannot really wait. So in the second arm of the project, what we have started doing is we are working with chemists here at Emory University. We have a great drug discovery team here. So we're working with chemists to synthesize new analogs, new molecules that look like the current Sumoylation inhibitor we are using now. So it will be our own. It will be our own that we can have our patent on, and then we can take those molecules to clinical trials and nobody can interfere because it will be ours. And so I'm going to update Keith on this matter. Actually, it's important that he knows about it, because this is the funding really came from CareStart now. So we're using this funding to generate some of this. They're called analogs. So they look like the drug we are using now, but with small modifications here and there in the chemical structure. And we are testing. We already got 5 analogs from our chemistry collaborators, and we are testing those 5 analogs as we speak now. So I just hired a new PhD student, Tare. She's excellent. She did her master's from UT Southwestern in one of my collaborators' lab, in a cancer metabolism lab, and she joined my lab as a PhD student. And she's going to be working full-time along with Skylar on this Sumoylation project. So she's now testing those analogs. So I think we have made great advances with the seed money that we received from the CareStart Foundation. And our goal is now to secure more external funding through other foundations, or mainly actually I'm going to write the first Sumoylation federal grant this fall. So that's where we stand now.
Wonderful. Also, can you speak to radiation? I know maybe in some of your
Yes,
work there
yes.
was benefit with mice with the inhibitor combined with radiation. Can you share a
Right,
little bit about that?
right. And so for the initial experiment we did with this one inhibitor, the one that is the bulky inhibitor that didn't really cross the brain very much, it did have an effect, and it did have an additive effect with the radiation. It was not very significant because the drug itself was bulky, so it did not penetrate the brain very much. So now we have the smaller drugs, but we just got fresh data like 2 weeks ago with the smaller drug, not in mice, but in vitro in three different cell lines, that at a lower dose of radiation, this small Sumoylation inhibitor enhances radiation sensitivity. So, for example, if the radiation will kill 50% of the cells, and if this inhibitor would alone would kill about, say, for example, 60, 70% of the cells, together they would kill like 90% of the cells. So that has happened in three cell lines in vitro on the petri dish now. And so now we're going to go in the animal, treat the animals with various doses of radiation and various doses of the drug to test if there is synergy, meaning that the drug and the radiation combination will work way better, whether it will work way better than either the drug or the radiation alone. Because, the standard of care is radiation. So what we'll propose is that once we radiate the patients, we'll treat the patients with this inhibitor, and we'll see if we can prolong, the survival of these patients with the inhibitor and radiation.
Mhm. Dr. Dasgupta, can you share what needs to be done from now until the first clinical trial can be available to patients? I'm assuming it likely would start with DIPG patients and potentially on the results branch out to other types, but what work still needs to be done in order for there to be a clinical trial?
Yeah. So luckily, we have a great team here, um, who have past and present expertise in running clinical trials in pediatric brain tumor patients. So that part is solid. I don't have to recreate that here. Children's Atlanta has an established protocol. They run clinical trials in various pediatric brain tumors. What needs to be done specifically for this project, um, is that we'll want to test if we can take the current HVO7 inhibitor to the two trial by working with the company. The path B is, as I mentioned, if we can come up with our own analog. But that needs to be, I think that route will be a little delayed because several toxicity studies have to be done in animals, and that's a pretty elaborate study. But I think the quickest way to get in clinical trial is to work with the company that has license of the small molecule, and we can go either directly inject to the tumor through catheters, through intracerebroventricular delivery, or using an ultrasound along with radiation. I think that's really the quickest path to clinical trials. I would say, if you ask me time for DIPs, I would say a year or two, maybe, if you can work with the company fast enough, and if they agree. But I think we should have the data ready, at least in the preclinical models. So one more thing that we do is, before we go, into animals, we also have organoid models. We create organoids. We have a collaboration at Johns Hopkins. So what she does is that she creates this organoids from stem cells, that looks like the brainstem. So this organoids, they look like brainstem, the human brainstem, and they mix those organoids with the DIPG cells, and they treat the organoids with the drug. Okay, and so in one shot we can know whether the drugs impact the viability of the normal cells in the organoid, because it's a normal brain, and how does it compare with the tumor cells, because the tumor cells are within the organoids. So this is like, as we call it, assembloid, meaning they're all assembled together. So it has got normal brain and the tumor cells growing as if they're growing in a patient or a mouse. And we treat everything. We treat the whole assembloid with various drugs, including the Sumoylation drug. So we do that. We also um, test another parallel method is we cut brain slices, mouse brain slices, and then we inject the tumor cells, DIPG cells, the brain slices, and we then treat those brain slices that contain the tumor cells, and we determine if the drugs have more effect on the tumor cells compared to the normal brain that surrounds the tumor. Yeah, so we are taking multiple approaches really, and so I would say if everything goes well, so me and Toby are really excited. Actually, we exchanged text this morning that how excited we all are as a group on this project. I would say I will really push it hard, so we can, have at least start working on a clinical trial in the next year or so. That potentially would depend on, if the company agrees or not. But at the same time, because the pathway is so important, we should be ready with our own analog at some point and in the near future.
Great. Well, it's really encouraging to hear all of this, and especially just the really global perspective everyone's taking, trying to address all the issues from all different sides of the spectrum. So we're very encouraged and excited to see where your research goes. I'd like to end our time with a couple of questions, kind of about the role of philanthropic support and the impact that it's had on your research. Dr. Dasgupta, I know many of our families spend countless hours organizing fundraisers and raising money for research. What would you want them to know about the impact that philanthropic funding has on the work happening in your lab?
Um, I would start by saying it's one word, which is gratitude. And I am saying this with a lot of weight in it. It's because it is their courage and their resilience and their determination that really inspires people like us to drive our work every day. Um, I mean, people may see that, especially patient families may see the uncertainty of today, but what I see as a scientist is the urgency of tomorrow. And um, and it is really this urgency that drives our work every single day. The main problem with in finding discovery for pediatric cancer, especially pediatric brain cancers, is it's very hard to get federal money, in general to begin with, because of the population size is small, and so there is always a thought, as wrong as it is, that, oh, even if a discovery is made, that it's only going to save 400 kids, as opposed to, let's say, for example, colorectal cancer or lung cancer is going to save millions of lives. For the same reason, pharmaceutical industry is not very interested. And so funding from organizations like Here Starts Now is a lifeline. Is a lifeline for the projects, is a lifeline for some of these projects that may, and I would say will, make a difference in DIPG or pediatric brain tumor research one day. I think the cure is probably laying somewhere in plain sight. It's just a matter of time. I think we're going to find it out. One of us will find it out. One of us in the DIPG community will find out. But it will all depend on continuous funding from organizations as dedicated as CareStats now. Because no one is going to fund us. No one is going to give us this $100,000, $200,000 seed money to run this research. To put it into perspective, we have another actually mega project also funded by CareStats now. I mean, we did so much with so little money, and it's the only foundation that funded us. It was through this $50,000 that was funded to our lab. Um, I remember that moment in Kentucky. There was a competition, this 5 minutes of presentation, and they received a check of $50,000. And with that money, we were able to screen 800 supposedly brain penetrating compounds, and we are now closing it. We have now closed down to about 10 compounds, and we are going forward with these 10 compounds in our mouse models. With that $50,000, we have done this much work. With that money, we have now collaborated with the artificial intelligence group here and asked them to tell us why these 10 compounds look better. And they came up with the hypothesis. They came up with the rationale. Their machine learning came up with the rationale. They ran all the 800 drugs that we screened through their machines. And then, and we now have through our collaborations with our AI labs, now we have specific drug combinations that we are going to test now. And so all of this happened because of the funding from CureSys now. All of it, like 100% of it. And so it's really, really important for foundations like CureSys. Now, of course, there are many other foundations that also help people like us through their philanthropy, through their work, through their determination. But it is this philosophy of funding labs with seed money to develop new projects. That is one day is going to lead to a cure of this lethal disease.
Very well said. Thank you so much, Dr. Dasgupta. It's been a pleasure talking to you today. Thank you for your time, your interest, and your perseverance in finding a cure. We really appreciate your time. Thank you.
Thank you, Rachel.